Hello, my name is Oren Shanks. I'm a senior scientist with the US EPA Office of Research and Development. And today I'm going to be telling you about a presentation entitled Moving Towards a Quantitative Human Fecal Source Identification Recreational Water Quality Management Tool. This presentation is organized into three parts. I'm going to start out briefly with an overview on what fecal source identification is. I'm then going to spend most of my time talking about draft EPA methods 1696 and 1697. And I will end with a short discussion on implementations and considerations. As many of you are well aware, fecal pollution is a nationwide challenge. Fecal microbes are actually the most common biological contaminant reported during water quality impairment. When fecal pollution reaches a certain level, it can become a public health risk, and it can also become an economic burden, especially in areas that rely on clean and safe water as part of their economy. Fecal pollution is typically monitored using a general fecal indicator. These are things such as E. coli or enterococci. These are microbes harbored in the gut and shed by most animals. So when you measure this in a water sample, it's giving you an estimate of the total level of fecal pollution. This information can be valuable because it tells us when there's fecal pollution present, and also if the level is at a point where it might signal a public health. What it doesn't do is discriminate between pollution sources. The source of fecal pollution can be very important. There's a growing body of evidence suggesting that public health risks can vary by different pollution sources. If your job is to mitigate fecal pollution, you know that you may handle a situation very differently depending on if the source of pollution is from a faulty septic system or line, compared to maybe dog access at a beach. So source information can help improve public safety and also improve water quality management. So in response to this need, the EPA has maintained an active research program in fecal source identification. And one of our most recent achievements is the public release of draft EPA methods 1696 and 1697. These are protocols that describe standardized human-associated fecal pollution PCR methodologies. These protocols are comprehensive, including information ranging from how to set up your labs, safety for handling all the reagents, detailed lists of all the equipment, reagents, and supplies that are needed, step-by-step instructions on how to collect samples, handle the samples, perform the qPCR testing. And in addition, there is extensive information on data analysis and data acceptance controls and how to do these calculations. In addition to that, there are also several companion tools that are available. There is a self-administered method proficiency test. There is a step-by-step procedure describing how to take and test these methods in your local area of interest to determine the performance in the hands of your laboratory. And there is also an automated data analysis tool, which is an Excel macro that basically will standardize and automate the data calculations. It will incorporate all of the quality controls and data acceptance metrics and generate an easy to understand report, including tables and figures. So for people that are interested in using this methodology, you can download all of these things at the website that's listed below. It's important to recognize that these protocols have been standardized for surface water quality applications. For 100 mil surface water volumes, all the reagents and controls are specifically designed to deal with environmental application. And it's important to realize that if you alter any steps in this standardized protocol, it could change the performance of the methodology. These protocols have also been confirmed in terms of performance through a national validation study, 16 participating labs where performance was evaluated with fresh and marine surface waters. In addition to that, we use this data to develop custom data acceptance metrics that are the backbone of the self-administered test that I spoke about earlier. Another thing that we've been doing is going out into the field and doing application demonstrations where we can show how to use these standardized protocols to solve real-world problems. A couple that I can share with you today are a large watershed scale monitoring exercise that was done in Tillamook, Oregon. This was done across a broad range of geographic space, 29 different sampling sites sampled over a year. How you can use these methodologies to understand seasonal trends and identify what's happening at particular sites using these methodologies. In addition, we've conducted and completed 2 studies in the Great Lakes regions focusing on recreational beach monitoring. In particular, how to use these technologies as companion tools with routine beach monitoring with a general fecal indicator. Another project that we're almost done with at this time is the development of a reference DNA material. We're partnered with the National Institute of Standard and Technology to get this completed. This reference DNA material is designed to work with draft EPA methods 1696 and 1697, as well as 11 other qPCR assays that are commonly used for water quality monitoring. This reference material is being certified by NIST using digital droplet PCR. So this material can also serve as a positive control for individuals that are interested in using any of these assays in a digital PCR platform. This will be available to the public this calendar year, and we're very excited to get this out as soon as we can. So for my last slide, I wanted to talk about some implementation considerations if you are a group that are interested in using this methodology. So a couple things that I would mention are make sure that you assemble a team of experts. Doing fecal source identification is an interdisciplinary endeavor. So you need people that know how to organize, run, and conduct molecular experiments. You need people expertise in the local area of interest in terms of what are the local regulations, where are all the sampling sites, what types of activities are happening on the adjacent land around those areas. And depending on what the situation may be, you might also need an engineer, you may need a wildlife biologist, or someone along those lines. Second thing I would say is when you're developing your study design, make sure that it's hypothesis driven. The more clearly you can define what question you're trying to answer and design your experiment specifically to answer that question, the more information you're going to get from those experiments. And that's going to lead to more focused, actionable outcomes after the study is complete. Another thing I would say is use the standardized method. As you can see, a lot of work has gone into standardizing and optimizing and validating these methodologies. In addition, I would recommend that you confirm method proficiency before water testing. That self-administered method proficiency test can be very valuable. It can be used to train people, but maybe even more importantly, it can be used to make sure that everything is working correctly in your labs, from the reagents, the instrumentation, as well as the analysts performing the tests. And then the last thing I would say is that technical support is available if needed. So there's experts like me in the Office of Research and Development, but there are also people that are familiar and have expertise in these methodologies in the Office of Water, as well as in our US EPA regional laboratories, which are stationed across the country. So with that, the last thing I'd like to do is acknowledge all of the people that were involved in making this method possible. There's a large number of us on the EPA side of the fence, as well as our external partners. We absolutely could not get this done without